Welcome, everyone. Thank you for your patience as we just sorted out a couple of technical difficulties, but we're ready to begin now. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. Good afternoon, everyone, and welcome to the May FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. Thank you for joining us this month. After a couple of international guests recently, we returned to our homegrown talent this month to hear about the exciting work being done locally. If you missed the April seminar from Dr. Ravindranath on endogenous retroviruses in ALS, or any of our previous seminars, the recordings are all available on our website now. Following the talk today, we are leaving plenty of time for discussion and questions. So if anyone would like to ask anything, please use the reaction button to raise your hand and one of our team will enable your microphone and camera and allow you to ask your question. Alternatively, you can also type your question to the chat where our team will be monitoring and read out questions on your behalf. Please note, however, that the chat function is only enabled if you are using the app-based version of Teams and unfortunately doesn't work on the web-based version. Please do ensure that you introduce yourself and where you're from before asking your question. So now to our speaker for today. Associate Professor Joe Nicolazzo is a faculty member and Associate Dean of Graduate Research at the Faculty of Pharmacy and Pharmaceutical Sciences at Monash University. After obtaining his PhD from Monash in 2004, Associate Professor Nicolazzo has established a strong research career, elucidating the fundamental processes that contribute to molecular transport across the blood-brain barrier and how neurodegeneration impacts on blood-brain barrier trafficking processes. He has been the recipient of FightMND funding for his studies and joins us today to discuss the results of this work. Joe, before you begin, could you let us know what you find most fascinating about MND research?
Yeah, sure. Thanks, Sarah. So yeah, I guess the most interesting and most inspiring thing around about MND research is the close association with the patient cohort and the families. It's I'm relatively new to MND research, and I've found that to be really inspiring and influential, and really gives us it makes it real. It makes research having the impact of the research real.
Yeah. And what would you say is your proudest career achievement to date?
So probably I would say back in 2017, so I had the opportunity to host an international blood-brain barrier conference, which brought together 220 guests from around 25 countries. And that was, I guess, being able to sort of bring all the international BBB experts to Australia and sort of welcome them to our country was probably one of the proudest moments in my research.
Great. Okay, well, thanks for playing along with my questions. I'll leave it over to you to start your presentation. Thanks.
Okay, so thank you, Sarah and to FightMND for the opportunity to present today. So as I did mention, relatively new to MND research, had more of a, experience with Alzheimer's disease and other areas of neurodegeneration, but was lucky enough to be invited to the MND symposium back in 2018, which was based here in Melbourne, and I was quite inspired by the area, and it gave me a real interest to sort of tackle some of the challenges in this research area, and I guess use some of my Alzheimer's disease knowledge and translate that hopefully into the MND space. So first I do need to acknowledge and thank FightMND for the support that they have provided over the past 2-3 years. It's been really helpful, without which we wouldn't have been able to do this work. So what I will do is describe a bit of a background to the blood-brain barrier and its importance and its role in trafficking of drugs across the BBB, and into the CNS, and also then focusing on how some of that changes in motor neuron disease and what impact that potentially has on drug delivery and medication safety in individuals with this disease. So I guess many of you may know that the blood-brain barrier is formed by the endothelial cells that line the cerebral capillaries within the brain. And so this is a vascular cast of a mouse cortex, and you can see that it's very well perfused with blood vessels. And in the human situation, it's been estimated that there are 640 or so kilometers worth of this vasculature going through the human brain. So it's quite a large network of microvessels, and the surface area that it provides for transport both into the brain and out of the brain is a key feature in ensuring that the brain homeostasis is well maintained. So if we take a transverse section, and Sarah, we can see the mouse. If I hope that's a yes. So
yes,
if we take,
yes, we can see the mouse.
Thanks, Sarah. So if we take a transverse section of one of those microvessels, we'll see the traditional view of the blood-brain barrier, or the neurovascular unit, which consists of these endothelial cells here in green, some tight junctions that seal these endothelial cells together, which I'll talk about in a minute. And there are some accessory cells such as the pericytes and the astrocytes, as well as this basement membrane around the endothelial cells and pericyte, which all form a very effective network to prevent certain molecules from getting from the blood, which is seen in the lumen, to the brain outside. Now, these endothelial cells, as I mentioned before, are associated with very tight junctions, which effectively seal the endothelial cells together, and that's a very effective network, and we can see a number of proteins there, such as occludin, claudin, and junctional adhesion molecules, JAM, all together, which, as I mentioned, effectively seal that intercellular space together. So if we make it even more simplistic and try and ask the question, well, how do molecules of any sort, whether they be drugs or nutrients, how do they get from the blood into the brain? And this is a very simplistic model. There's two possible routes. There's the paracellular route between the endothelial cells and the transcellular route across the endothelial cells. Because of the presence of the tight junctions, at least in the healthy conditions, the paracellular route of transport is effectively sealed, and so no molecules can traverse the paracellular route and in the intact and healthy state, which leaves the transcellular route. So molecules can only cross an effective BBB, blood-brain barrier, if they are able to cross in some way through that endothelial cell. For small lipophilic molecules, that's quite easy because they're able to basically diffuse through the endothelial membrane and be trafficked across. And in general, the smaller the molecule, the more lipophilic the molecule, the greater is its ability to be absorbed across the blood-brain barrier via a simple passive diffusion process. And so this is shown in this graph, which we generated over 10 years ago, where we looked at the brain to plasma ratio of a whole series of radiolabeled drug-like molecules. And what we did is we IV administered these molecules into mice, healthy mice, and after 5 minutes looked at the brain to plasma ratio, radioactivity in the brain relative to radioactivity in the plasma. And what we can see for the compounds in blue, there's a nice relationship between log D, lipophilicity, so this is basically a measure of fattiness of the molecule, and brain uptake. The more lipophilic the molecule, the more is going to be its brain uptake. And that follows generally a nice linear relationship. And if we look at the properties of these drugs, they all permeate via simple passive diffusion. So this is a nice, what medicinal chemists would like to do is make sure that their drugs fit on this line, make up their drugs to have the right properties to fit on that line, which is all well and good, but oftentimes what we see is a bunch of molecules that, while they have the right lipophilicity, the right log D, their brain uptake is much lower than that line, that passive diffusion line. And the reason for that is because these drugs are substrates of efflux transporters. So the diagram I showed on the previous slide, it's not as simple as that, because what we have are these efflux transporters such as P-glycoprotein, PGP, breast cancer resistance protein or BCRP, and MRPs, multi-drug resistance associated proteins. And what these proteins do is that they identify molecules that are generally lipophilic, and they basically suck them up and kick them back out into the blood. So you might design a molecule that has good lipophilicity, think it's going to get across that membrane, but it's recognized by a transporter like PGP, which effectively kicks it back into the blood. And so these molecules would have a much lower brain uptake than what you would predict based on their lipophilicity. And these transporters, therefore, are a really, I guess, an additional barrier, a biochemical barrier to drug delivery, because paracellular diffusion is not possible if the tight junctions are there. And so we are reliant then on simple passive diffusion for molecules to get across, but then these transporters come in and potentially efflux our molecules back out. But it's not that bad. There is some good news. And there is some good news because we know that endogenous molecules like glucose, amino acids, and so forth, they need to get into the brain, and they don't have the right physicochemical properties or lipophilicity to get across. And so what we have are basically carrier-mediated transporters that assist in trafficking of molecules like that into the brain. So we have LAT1, which is an amino acid transporter, GLUT1, and also some larger receptor-mediated transporters that I won't speak about today. And so these molecules are going to, or molecules that utilize these influx transporters rather, would have a higher brain uptake than what you would predict based on their lipophilicity. And that's exactly what we see in our graph here, where we look at the brain uptake of substrates that are, or molecules rather, that are substrates of these transporters. They have a greater brain uptake than what you would predict based on simple passive diffusion, i.e. that blue line. So that's a very basic overview of simple blood-brain barrier transport processes to hopefully set the scene and give you some insight into what is important before we move on to MND. When we consider MND, and I won't go through the pathology or the genetics because I know most of you are all familiar with that. What I'll do is I'll jump straight into what is known about the blood-brain barrier or the blood-spinal cord barrier in MND, or in the majority of the studies in ALS, the subset of MND. And so it's been known for many years that the tight junctions between the endothelial cells are reduced in expression. So here's some data that's looking at human spinal cords in non-MND individuals, and then individuals with either sporadic or familial ALS. And what we see here, Z01, zonular occludens, one of the tight junction proteins, the expression is significantly reduced relative to non-ALS. And similarly for occludin, in familial ALS, there is a significant reduction in the ALS individuals relative to the non-ALS individuals. And so the tight junctions are open, the paracellular route is open, and therefore, as a result, we would expect there to be leakage of a lot of brain-related material into the spinal cord and perhaps the brain as well. And in fact, there have been some studies, again, in patients or individuals with ALS where hemoglobin levels have been measured in the cerebrospinal fluid of individuals with and without ALS. And what this graph is showing, and this is a fairly recent study, is the concentration of hemoglobin in the CSF in control and in individuals with ALS. And what's generally seen is that at higher concentrations of hemoglobin, there is a much greater proportion of, or greater readout for those with ALS relative to those without ALS. And so this is in line with the suggestion that, or the data that tight junctions are lower in expression, and therefore there is a leakier barrier, and therefore large molecules like hemoglobin can then enter into the CSF. But we need to remember this is CSF, it's not parenchymal tissue, and this is not necessarily, the increase in CSF levels of hemoglobin is not necessarily only due to tight junction dysfunction, but could be due to changes in the blood CSF barrier. So we just need to keep that in mind. When we go to mouse models, so I've got some data here from another lab from some time ago, where they've looked at spinal cord microvessels from the SOD1 G93A mice, the mutant model of ALS. And for a number of tight junctions, so zonular occludens 1, occludin, claudin 5, there is this reduction in, this was mRNA expression of the relevant tight junctions. And I say may be reduced in the title because there's other studies that have reported no change to the mRNA and the protein expression. So I think it's very much dependent on the age at which the studies are occurring, and also which regions of the spinal cord or the brain are being assessed. But if there is a reduction in the tight junction expression, what you might expect to see is, as the case in humans, a leakiness of that barrier into the spinal cord. And this indeed has been shown by others, where we have some slices of spinal cord of mice that were either wild type in panel D or SOD1 in panel H, and that were administered a dye, Evans blue, and you can see that in the wild type mice, the microvessels are very well structured, i.e. the dye is remaining within the microvessels where the junctions are allegedly intact, whereas in the ALS mice we see a large, I guess, blurring of that dye, which is suggesting that the tight junctions are indeed disturbed, and we're getting leakage into the spinal cord. So based on this, you would expect that drug penetration into the CNS, whether it be the spinal cord or the brain, should be increased in ALS. And that, I guess, has generally been dogma, as it is in Alzheimer's disease and in Parkinson's disease. But this may not actually be the case. It may be that there is either no change or a reduction in CNS uptake of drugs. And so, looking in the literature, there was an interesting study that assessed the spinal cord uptake of this molecule, LD800. So this is a probe molecule. It's not necessarily a drug. But after IV injection, if we look at the spinal cord levels in the wild type on the left and in the SOD1 mice on the right, we actually see a significant reduction in the spinal cord uptake of this particular molecule. Similarly, with digoxin, another study looked at the brain to plasma ratio of digoxin in wild type mice and in SOD1 mice, and again, a reduction in the spinal cord uptake of digoxin. So why is this occurring? If the tight junctions are open, shouldn't we expect to see drugs getting into the CNS to a greater extent rather than lower extent? Before I go into the reason why, we need to appreciate that these two drugs, well, digoxin particularly, and this probe molecule, are actually substrates of PGP, this efflux transporter. And so what was suggested by this was that perhaps PGP function or expression was actually increased, and that was what is resulting in a lower uptake of these particular PGP substrates into the spinal cord. And in fact, that's actually the case. PGP and BCRP, the two efflux transporters, are actually expressed at a higher level in ALS spinal cords and also in brains relative to control. So here we have spinal cord homogenate, which has come from humans, and the bands for PGP and BCRP, you can hopefully appreciate, are much stronger than in ALS relative to the control setting. This has also been seen in a mouse model. So again, this is the SOD1 mouse model, and what I'd like to show you here is these are the microvessels that are being probed for von Willebrand factor, an endothelial marker, and this is PGP. In the control, quite light; in the ALS, quite high. So it looks like both in humans and in mouse models, there is this increased expression of PGP, which is likely increasing the efflux of molecules from the brain back into the blood, therefore lowering CNS uptake of molecules. What's also interesting is that riluzole, so the one therapeutic that's, approved by the TGA for MND in Australia, is also a substrate of PGP. And this is demonstrated by this particular graph where the percentage of riluzole in the spinal cord or in the total CNS is compared in wild type versus PGP deficient mice. And so when mice lack PGP, there is a significant increase in the spinal cord or total CNS uptake of riluzole. When we take into account that riluzole is, when we take into account the fact that riluzole is a substrate of PGP and PGP expression is upregulated in ALS, perhaps there are ways that we can start manipulating PGP, inhibiting PGP, so that we can get more riluzole into the brain and the spinal cord in order to have a better therapeutic effect. And this has been done using alacridar, a PGP inhibitor, and you can see that that increases the uptake of riluzole into the spinal cord quite dramatically in healthy mice. And these investigators went one step further to see whether inhibition of PGP can increase the survival of ALS mice, or SOD1 mice, I should say, with and without, with a PGP inhibition with alacridar could improve survival of these mice that are taking riluzole. And you can hopefully appreciate, while there was some increase, it wasn't dramatic, and therefore lifespan wasn't enhanced to a dramatic effect. And similarly with the grip strength, we see that the presence of riluzole reduces the deterioration in grip strength. So, in the presence of alacridar, I should say. So PGP inhibition can be one way to improve riluzole uptake into the CNS. However, the doses that are required in the clinic in order to inhibit PGP are actually quite high, because this has been tried for many other diseases, and it comes with a lot of off-target effects. And so while this might appear promising, I think there's quite a bit of work that needs to be done to sort of modulate PGP in other ways if we would like to increase riluzole uptake. So I kind of went on a little bit of a tangent there with the riluzole, but I thought it was important. But hopefully you're starting to appreciate through this presentation that there is an alteration to the BBB and the blood-spinal cord barrier in MND. And that may not only provide us with targets that we can potentially modify to improve therapeutic delivery or delivery of our therapeutics into the CNS, but it also begs us to ask the question, what about all the medications that individuals with ALS are taking for both their MND symptoms, but also for the other conditions that they may also be diagnosed with, such as high blood pressure, diabetes, cholesterol, etc., etc. We dose individuals with MND at the same dose as otherwise healthy individuals. If the blood-brain barrier transport of these particular drugs is being modified, then we may be seeing subtherapeutic or supratherapeutic concentrations of drugs in the brain and spinal cord, which may impact on the patient's well-being and side effect profile. So in order to, get a better understanding of what the BBB is like in MND, we did a systematic study where we looked at the brain uptake of a series of molecules in the one lab in two different mouse models. Firstly, the C9 or FBAC mouse model, and secondly, in the SOD1 mouse model. And in these wild type and MND mice, what we did is we used the transcardiac perfusion technique, which we often use in our lab. And what this technique involves is opening the thorax of the mouse, ligating the descending aorta, injecting our molecule of interest through the left ventricle, which then perfuses the brain and leaks out of the severed jugular veins. And so it's a one-way perfusion at a rate of 10 mils per minute for a 1 minute period at a known concentration of probe drug. And so what we did is we assessed a number of probe drugs in both wild type and C9 mice, and then later in wild type and SOD1 mice. and we determined the brain to perfusate ratio of each of these drugs to get a feel for what is actually happening to the blood-brain barrier in these particular mouse models. And I've highlighted their BBB because we only looked at brain. Unfortunately, we didn't look at spinal cord due to sensitivity issues. So just we need to keep in mind that this is the true BBB as opposed to the blood-spinal cord barrier. And again, I need to acknowledge FightMND for the support that allowed for Yijun Pan, who was the postdoc, who was supported by FightMND in order to undertake these studies. So, in order to systematically characterise the BBB, firstly we used the C9 back mice. The first compound of interest was sucrose. Sucrose is a marker of the paracellular space, and it gives you an indication as to whether the tight junctions are actually disturbed or not. And so the brain to perfusate ratio of sucrose in wild type and C9 mice was no different. If the tight junctions were significantly disturbed, we would expect to see a much higher brain to perfusate ratio in the C9 mice. Just to show you what it looks like when the BBB is disturbed, these are some studies from, some results from another study we've undertaken in the past, where we've intentionally disturbed the blood-brain barrier by administering LPS to healthy mice. And you can hopefully appreciate there when we've intentionally disturbed the BBB, there is a significant increase in the brain to perfusate ratio of sucrose. However, in the C9 mice, no apparent disruption to the BBB in terms of tight junctions. We then looked at diazepam and caffeine, two passively diffusing markers, one quite lipophilic, diazepam being quite lipophilic, caffeine being quite hydrophilic. And we didn't see any difference to the brain to perfusate ratio of either of those two compounds, suggesting that the BBB, in terms of passive diffusion, appears to be similar between the two mice. We then looked at digoxin. Remember, digoxin is a substrate of PGP, and if PGP function or expression is increased, we would expect to see lower brain uptake of digoxin. But actually, we saw no change, no significant difference in the digoxin brain uptake. So far, the studies from this mouse model were suggesting that, or were not concordant, I should say, with what we were expecting. Finally, we looked at three influx transporter substrates: alanine as an amino acid substrate for the amino acid transporter, oleic acid, fatty acid transporter substrate, and 2-deoxyglucose as a GLUT1 transporter substrate. No change to alanine, no change to oleic acid, but what we saw was a very interesting finding, an increase in the trafficking of 2-deoxyglucose across the BBB. This was not due to leakiness of the BBB, because we'd already shown that sucrose was no different, and all of our probes were no different in terms of trafficking, and so it appeared to be a substrate-specific effect. So what Yijun then did is isolated microvessels from wild-type and C9 back mice, and then undertook an ELISA to see whether the GLUT1, the glucose transporter expression levels were increased. And actually they were. There was an increase in GLUT1 expression, which was associated with the increase in glucose trafficking, suggesting there was more glucose getting into the brain. And at the time, this really confused us and concerned us, because we know in ALS, MND, there is actually glucose hypometabolism. And so there's less glucose being metabolized, and so why would there actually be more glucose getting into the brain in this mouse model? So we kind of put this to the side and thought, let's look to see what's happening in the SODs. SOD1s being a more, I guess, well-established model, perhaps that may give us similar or different answers. So we then moved to the SOD1 mice, and we undertook similar studies to what we did with the C9s, or the C9 backs. And so I'll get straight into it. Sucrose, paracellular marker. If we're expecting tight junctions to be disturbed, then we should see more sucrose getting into the brain. And this is where it really got interesting, because what we actually found is that the paracellular route, or the penetration of sucrose, was actually lower in the SOD1 mice relative to the wild-type mice. Very interesting finding. perhaps in the SODs that we were using, the tight junction expression was not lower. And so what Yijun did was isolated the brain endothelial cells from wild type and SOD mice, and through a proteomics approach looked at a number of tight junction proteins. And what we can see here, wild type versus SOD, is that a number of these tight junction proteins, including occludin, were significantly reduced in the SOD1 brain endothelial cells. So if there's reduced expression of the tight junctions, shouldn't we see more sucrose getting across? We should, but I guess the blood-brain barrier is not just formed of endothelial cells and tight junctions. As I mentioned previously at the start, there is the basement membrane that surrounds these microvessels. And so we wondered whether microvascular basement membrane was thickened in these SOD1 mice, and whether that may be causing some sort of reduction in the ability for sucrose to potentially get across. And so Yijun stained these microvessels, or stained the slices rather, with collagen 4, which is a basement membrane protein. And what we saw was there was an increase in that thickening in that basement membrane, and potentially also a shrinking of the luminal diameter. And so perhaps the reduction in sucrose penetration might actually be a result of basically constriction of those microvessels, a reduced vascular volume. This is not the first time this has been observed in ALS. There was a study that looked at human spinal cord collagen 4 deposition, and what they also found in the human setting was that the basement membrane was also thickened in the spinal cord. So this was an interesting finding. It kind of went against the dogma that perhaps brain penetration of drugs is always increased, and at least for sucrose, it was reduced. We then went to look at diazepam and caffeine, and we saw similar findings for diazepam, not for caffeine though, which was in itself interesting. And there was about a 40% drop in the trafficking of diazepam across the blood-brain barrier in the SOD1 mice. And we've actually seen exactly the same results in a mouse model of Alzheimer's disease, where diazepam penetration was reduced significantly, but caffeine penetration was not reduced significantly. And we think that that's got to do with the lipophilicity of diazepam. If there is a thickening of the basement membrane, which is mainly protein-based, that may form more of a hindrance to a lipophilic molecule like diazepam, as opposed to a hydrophilic molecule like caffeine. Regardless of the mechanism, which we don't fully understand, it's interesting to note that there does appear to be a reduction in the trafficking of a very well prescribed and utilized medicine across all patients. We then looked at digoxin. Remember, digoxin is the PGP substrate, and we would expect that if PGP expression or function is increased, then we would get less digoxin trafficking into the brain. And that's exactly what we observed. We saw a small but significant reduction in the trafficking of digoxin across the BBB, which we attribute to increased PGP function. You might ask, well, isn't it possible that the thickening of the basement membrane may be responsible for this reduction in digoxin permeability, like I just said for diazepam, which is possible. So in order to address whether this was a brain endothelial specific event and not a result of basement membrane thickening, what Yijun did was isolate the brain endothelial cells from the wild type and the SOD1 mice, and then looked at the uptake of digoxin into those endothelial cells, so that we were looking at a cellular uptake process in the absence of basement membrane contribution. And what he saw there is, in the SOD1 mice, there was a significantly lower accumulation of the digoxin in the endothelial cells, which is in line with increased PGP function, increased efflux ability of that transporter. So it did appear in the SODs, unlike in the C9s, that there was increased functionality of PGP, which was lowering the uptake of digoxin, and likely be responsible for the lowering of ribosol uptake as well into these brains. And finally, we looked at GLUT1 function. So remember, with the C9s, we had that weird observation, or interesting, I should say, observation, that there was increased glucose trafficking across the BBB. We actually saw the same thing in the SOD1 mice, increased trafficking of glucose. So this suggested to us that perhaps the findings we observed in the C9s were real, and that it did translate across to this additional mouse model. How this relates to the glucose hypometabolism, that's been very well reported in the literature in ALS. I'm not sure, and really interested to hear thoughts on that from the ALS community. But it's an interesting finding, and it may be an opportunity to, I guess, exploit this transporter to improve drug delivery across the BBB in ALS. So if there is more GLUT1, are we able to modify the structure of our drugs, make them appear more like glucose as a way of smuggling them across the BBB? So that's one thing that we're interested in looking at. The final thing that I wanted to touch on, and in the last few minutes, was trying to associate the increased GLUT1 function to the PGP overactivity. And why would I say that? PGP is an ATP dependent cassette binding protein. So it utilizes the energy from ATP to basically function. When there's more ATP, there is going to be a greater function of PGP. So we were thinking, is this overactivity of PGP somehow associated with the increased glucose trafficking, the increased potential energy production within the endothelial cells? And if we somehow block that, could we restore PGP function? So to address these final questions, what Yijun did is again, he isolated the endothelial cells from the SOD mice and the wild type mice, and used JC1 staining, which is an indicator of mitochondrial function. And what he observed in those endothelial cells, brain microvascular endothelial cells, is indeed an increase in mitochondrial function, suggesting ATP production. And in fact, he used the seahorse assay, which is an assay that allows you to measure the production of ATP. And in those SOD1 brain endothelial cells, he was able to demonstrate that there was actually increased ATP production in the SOD1 endothelial cells relative to the wild type endothelial cells. So, given that there was indeed increased energy production or increased ATP production, what we were speculating was that the increasing glucose trafficking into the endothelial cell is resulting in, through glycolysis, increased ATP production, and that increased ATP production was assisting to drive the increased function of PGP. So what Yijun thought in the final experiments was, let's try and block glycolysis. If we block glycolysis and have less ATP production, then we may be able to reduce PGP hyperactivity or PGP overfunction. And so to do that, Yijun used 2-deoxyglucose at fairly high concentrations in order to block glycolysis. And then he looked at the uptake of digoxin in the presence and absence of this glycolysis inhibitor. So this is actually the data I showed you before. The uptake of digoxin into wild type endothelial cells and into SOD1 endothelial cells, and we see less digoxin going into the endothelial cells from SOD mice because we have PGP overfunction or overfunction of PGP. In the presence of 2-deoxyglucose, however, we abolish that effect. There no longer is that difference in PGP function between the two types of endothelial cells. So I'm not saying that we should inhibit glycolysis as a way of restoring PGP function or bringing PGP function back to normal, but it does open up the suggestion that there are ways that we could, I guess, intelligently modulate PGP function or overfunction as a way of restoring PGP back to normal, so that in individuals with ALS, there is that greater ability for drugs to enter into the brain to exert their therapeutic effects. So as a summary, hopefully I've been able to, I guess, give you some insights into possible changes that may be occurring at the blood-brain barrier, particularly the increase in glucose transportation, which may be effective for improving drug delivery, the increase in PGP expression and function, which might actually be a problem for CNS delivery, but something we may be able to modulate, and also a thickening of the basement membrane, which, while important from, I guess, a pathological perspective, may also be reducing CNS penetration of therapeutics. And so maybe we need to consider avenues for modifying the doses of drugs we want to get into the brain in individuals with ALS, because there may be these additional BBB specific events that we need to consider. And maybe there are also opportunities, such as through the GLUT1 transporter, that we can more effectively deliver our drugs of interest into the brain, which may require some clever chemistry in order to ensure that our drugs can interact with the glucose transporter, but at the same time don't lose their efficacy or their binding affinity to their target of interest once they get into the brain. And last but definitely not least, I'd like to acknowledge the current members of the lab listed on the left-hand side, as well as some previous members who have contributed to this work. But definitely need to acknowledge Yijun Pan, who did all of the ALS work, and there's Yijun down the end there. He's actually now working at the Florey. Also, the Monash Proteomics and Metabolomics facility for their assistance with their proteomic data generation, and Brad Turner and Brittany Kewick from the Florey for providing us with the breeding pairs for the SOD and C9 mice, and also for providing a lot of advice and suggestions for these particular studies. And last but definitely not least, FightMND for the financial support of this work, and also for being great partners in helping to shed some light on this disease. And with that, I'm happy to pass it back over to Sarah and take any questions.
Thanks, Joe. That was a really interesting presentation. I'm going to moderate the questions now, so I open the floor up to questions. If you'd like to ask a question, you can unmute yourself and turn your camera back on if you choose, and just raise your hand and we'll unmute you so that you can ask your question. We ask, if you are going to ask a question, to please introduce yourself, just your name and your affiliation, just so that we can all get to know each other a little bit better. While we're waiting for our first question, Joe, I wanted to ask you about, I know that you've shown the increase in glucose transport and an upregulation of GLUT1 transporter. I just wonder, what's your thoughts around what might be driving that increase in expression?
Yeah, that's a very good question. It's something I don't have the answer to. I've been considering that if there is this hypometabolism that's occurring within the brain parenchyma, that the BBB, being the gatekeeper to the brain, is somehow trying to compensate for that, and as a result is increasing or trying to increase the amount of glucose that is getting into the endothelial cells. I know that PET imaging has, with FDG, has shown that there is actually lower glucose metabolism, suggesting lower uptake, but maybe there is increased uptake but then lower metabolism. It's something we need to look into, and it's something definitely was unexpected.
Mm-hmm. I thought I'd throw you a tricky one first up.
Thank you.
But it's really fascinating, and I think I know at FightMND, what we loved about this project was that thinking about it from a patient perspective and the other drugs that they're on, and how that the changes in the blood-brain barrier might impact that. So really fascinating research. We've got a question from Danny Hatters. I'll just allow your mic, and Danny, if you could just introduce yourself as well.
Can you hear me okay now?
Yes.
Yes.
Oh, great. Yeah, I wasn't sure if my camera was on. So I'm Danny Hatters. I'm at the University of Melbourne. I'm not sure if my camera is working. Yeah, I wanted to follow up a little bit on Rebecca's question. And I'm very naive about the blood-brain barrier biology, but I guess given that it plays such a crucial role as a barrier between the blood and the brain, I'm just wondering whether you can comment on what the risks might be. You know, for a therapeutic strategy that might be targeting that permeability, I can imagine there could be lots of potential challenges. I'm just wondering whether those challenges are well understood or whether that's something that you've been looking at as well.
Yeah, it's a really good question, particularly, I guess, at a time where researchers are trying to, I guess, open up the BBB using approaches to sort of, disturb the tight junctions and allow molecules to get in. I think that can come with some risk. And I do think that those sort of very generalized approaches perhaps should be taken with care, particularly for a long-term disease such as ALS, MND, or Alzheimer's disease, where sort of this constant opening of the BBB may lead to long-term effects. So I think any approach that reduces the barrier nature of the blood-brain barrier, if I could be quite bold, should be avoided. So even inhibiting PGP, inhibiting BCRP, I think they're there for a reason, and I don't think we should really play around with them. What I think we should probably do is utilize those influx transporters to our benefit and understand new ways to try and, as I mentioned before, smuggle cargo through the BBB, and making sure that that's not at the expense of preventing the nutrients, the substrates of those transporters from getting into the brain.
Yeah. So just, are there sort of assays that are established that can sort of evaluate negative effects? I mean, I'm not sure whether there are particular phenotypes that you might expect to see if there are problems with the blood-brain barrier permeability.
Do you mean from sort of just assessing if the BBB is perturbed, or do you mean long-term effects of? Yeah,
well, I'm thinking more about long-term effects. You know,
yeah,
just for example, you have a treatment that increases the permeability. What happens long-term? Are there assays or ways that we can sort of track potentially detrimental things?
I think, so from an in vivo standpoint, probably looking at sort of microgliosis, activation of astrocytes, basically any sort of neuroinflammatory insult or increase would probably be my suggestion as a readout, that things that shouldn't be getting into the brain are getting into the brain.
Great, thank you.
Great, thanks, Danny. And we've got a question from Rebecca Nisbet.
Hi, Joe. Great talk. I was just wondering, have you ever seen, in particularly with your SOD1 mice, have you ever seen inclusions in the brain endothelial cells?
No, but we haven't looked.
Okay.
So, no.
Yeah, because we see some funny things in some of our tau models, and I'm wondering if that also prevents drug uptake if you're getting protein-specific inclusions in the brain endothelial cells and it's kind of getting trapped in the vasculature.
It's quite possible. Yeah.
Okay. Thank you.
Sorry, I couldn't answer your question.
No, that's all right.
If we haven't looked at that, no.
Great, thanks, Rebecca. Joe, just a sort of general question. Obviously, you're an expert in this area, and there's a lot of people who work in MND and in sort of development of potential new treatments or therapeutics. What's some advice you can give if you're looking at designing drugs or developing drugs about how to ensure that researchers are looking at the capability to cross the blood-brain barrier?
I guess where possible, assessing transport as early on in the drug development pipeline would be one piece of advice, because it's too often we can get really excited around some in vitro work and have activity and efficacy, etc., or activity in vivo through an ICB injection, but it's kind of delaying the inevitable question of, does it actually get into the brain? So I think assessing that as early as possible would be my piece of advice, because been collaborating a bit with others, and it's like, it's quite sad after all the sort of years of work that researchers have done to then sort of say, actually, no, it doesn't get across. We've done some tests and it doesn't get across. So yeah, that would be mine. And if there was sort of interest in small molecules, I would also recommend assessing the general oral absorption, or PK, as early on as possible, because that can really guide how much we dose and how often we dose. And particularly with ALS, it's something I didn't touch on because I didn't have time, but not only are we seeing blood-brain barrier changes, but we've got some really exciting data now that's showing that oral absorption is actually modified in the SOD1 mice, which then opens up another big question about just general dosing of any medicine in MND.
Great, thank you. That's so fascinating. Any more questions from the floor?
Well, I might leave it there then if there's no other questions from the floor. Thank you so much, Joe, for a wonderful and informative presentation and a fantastic discussion session. It was really great to hear about some really recent data and how it's currently being driven along the development pipeline. So if you do think of any other questions after this session and you want to follow up with Joe, please get in touch with me and I can connect you. I'm sure plenty of people might have questions about how they can collaborate with you on progressing their drugs through the development pipeline and ensuring that it does cross the blood-brain barrier. So today's session has been recorded and will be available on our website in about a week's time. So if you know of your colleagues who weren't able to join live today, just let them know that they can watch it soon. We have a bit of a change of direction coming up for our June seminar. Instead of a research seminar, you'll be hearing from our very own Director of Cure Research and Programs at FightMND, Dr. Beck Sheehan. FightMND is implementing a new research strategy over the next few years, and this will affect how our research investments are made. Dr. Sheehan will be outlining both our cure, research and care strategies and some of the key changes that you will start to see coming up. So if you'd like to hear about FightMND's new strategy, please tune in on June 27th at 12 p.m. to hear all about it. Until then, we'll see you then, and bye for now.